Today we are continuing our discussion of aseptic technique, this time in regards to inoculating auger plates.
The next important aseptic technique is inoculating an auger plate. Auger, which is made from seaweed, provides a growth medium for microorganisms. An auger plate is made from pouring warm auger into a petri dish. We've added nutrients, which is a microbe's version of steak and eggs, to the auger, so that they will grow and form a colony which is visible to the naked eye.
Today we are inoculating some microbial cells we harvested from soil onto auger plates with glyphosate. First, label the base of the plate with a small sticker or permanent marker near the edge. Be sure to include the date, the name of the medium, the name of the organism, and your initials. Take your sterile inoculation loop and insert the sterile end into the liquid culture containing the microorganism, or touch a colony on a plate. Now that you have the microbes on the loop, remove the lid on the new petri dish that you wish to inoculate by slightly lifting one edge away from your body. Keep the petri dish open for a minimum amount of time to reduce the risk of airborne contamination. Next, gently rub the loop on one section of the plate. When you do this, you are depositing millions of microbes on the auger surface. Try not to gouge the auger. Close the lid and flame the loop again. Allow it to cool, and now pass the loop over the area that you just inoculated and drag the loop into a new section of the plate. Repeat this procedure multiple times to inoculate the entire plate, following the procedure shown in this image. It is important that the auger jelly remains in one piece so that the culture has a smooth surface to grow. The lid should then be placed back down. You may want to seal the plate with tape or parafilm. Be sure to turn the plate over so that when incubating, the condensation does not drip into the growing culture. To sterilize the inoculation loop, it should then be flamed again. And the auger plates should be placed in the incubator at the appropriate temperature.
Another way to inoculate an auger plate is to use the spreader plate technique. Here we are inoculating a yeast solution onto an auger plate. First, ready the auger plate. Then take the pipette and attach a sterile tip. Adjust the dial to the exact number of microliters desired. In this case, we want 100. Draw up 100 microliters of yeast solution. Make sure to remove the lid with your left hand and hold it there while you draw up the solution. When removed, add the cap back to the lid. Then, while holding the pipette in your left hand, dip the glass spreading tool in ethanol and flame it. Inoculate the auger plate. Dispose of the sterile tip into the biohazard bag and begin spinning the spreader plate. Next, use the glass tool to spread the yeast along the plate. Make sure to only use light pressure and move the tool up and down. This ensures even distribution along the plate. When finished, make sure all equipment is clean, sterile, and returned to its proper place.
Don't forget to properly use these aseptic techniques in the biology laboratory when necessary.